Do you hear me? Okay, it's fine. Now it works? Ah, there we go. Now it works. The power button always helps. All right. Well, welcome, y'all. Full house tonight. Thanks for coming to see Bobbie. We really appreciate it. So my name is David. I'm the campus coordinator here at Reef. Thank you so much for coming. This is our Fish and Friends event. It's the second Tuesday of each month from January to August. So we've got 2 left this year. Next month will be Tom Matthews speaking about lobster and kind of lobster populations in the Keys just in time for mini season. And then in August, we'll have Stacey Henderson talk about underwater photography. So if you're interested in, yeah, so exciting. A couple things we've got coming up at Reef. We've got a Fishnar, which are just our online fish ID classes. They're also recorded. So if you go online, you can see this huge database of free fish classes for pretty much any destination in the world, which is awesome. We're heading to Roatan soon. So I'll be talking a little bit about some of the fun fish you can find in Roatan. So if you're interested in going to Roatan ever, it's a great class to tune into. We have the great annual fish count through all of July. We kind of take that month to focus on counting all of the fish that we can and encouraging dive shops to teach a fish ID class and go out and survey some fish. So if you're interested in being a part of that, or if you're at a dive shop and you'd like them to be a part of it, talk to Sierra, who's back there in the red, and she can tell you some of the events we've got planned already and some ways you can get involved. The other thing is Reef Fest registration is now open, so that's very exciting. If you haven't signed up for Reef Fest yet, you should definitely do that. That's kind of like a Reef family reunion that's hosted here in Key Largo every October. So we'll have a couple days of diving, snorkeling, speakers from around the world talking about whatever research they may be doing, and then we'll cap it off with a celebration on Saturday night. And then on Sunday morning at Pennicamp, Lori's giving a tour of the butterfly garden and the nursery, which will be awesome. All right. And last but not least, thank you to everybody who signed up for the Reef 5K. I see everybody's wearing their shirts tonight. A couple people are. Good work. We had 501 registrants. That's what we ended up at. So excellent work, everybody. That really helps fund the Ocean for All program, which allows us to deliver classes to anyone who is interested at little to no cost, which is super helpful. All right. Let's see. So another thing that's coming up is our Lionfish Derby and Festival in September. The registration is now open. This slide says a lot of things about it, but really what you need to know is if you are interested in spearing lionfish, you should sign up for our Lionfish Derby. It's $75 for a team of 4, and you get 4 super cool rash guards just for entering. And we'll do prizes for biggest, smallest, and most. And there's also 2 divisions. So if you see those pictures with hundreds of fish all over and you're like, wow, I don't think I can get hundreds of fish, don't worry. We have a division for you too, the entry division. Yes. But regardless, there will be a festival on Sunday, September 10th. So definitely stop by, even if you're not a diver, even if you don't like spearing fish. There is some really cool stuff we'll have going on at Postcard Inn, which is down in Isla Mirada. There will be vendors, there will be booths, there will be all sorts of neat things that you can do. So definitely bring the family, bring everybody to that one. That'll be a Postcard Inn down in Isla Mirada. Yeah, absolutely. All right. And then we're just about to wrap up our Lionfish Sweeps, which is kind of like our pre-tournament for the Lionfish Festival. We've collected over 150 lionfish so far. So good work to everyone in the community. I know there's some people down in Isla Mirada who've been really going head to head. So it's pretty close between Marilyn Stedman and Cassandra Clark right now. So I don't know if anybody on the Key Dives boat knows about that, but it's tight. So we'll start to post soon about Rock the Dock at Sharky's. They're still determining a date for that, but we'll let you all know it's a night that kind of benefits Reef and encourages the local community to come out together and enjoy a great plate of barbecue and support Reef. All right. And without further ado, introducing, yes. What's up? Oh, yeah. We also have our summer interns here, so they want to come on up for a second. Yeah, come on up. Yeah, all right. Woohoo. All right, so I'll let them introduce themselves and say where you're from.
All right. Hello, everybody. My name is Brian Chansky. I'm originally from just outside of Boston. I go to school at the University of Delaware. I'm going to be a senior, so got one more year left to finish, but I'm studying marine science.
I'm Carolyn. I just graduated from the University of San Diego, but I'm originally from Los Angeles area. and I was studying environmental and ocean sciences. Yeah.
All right. So Brian and Carolyn will be here for the next 3 months until the middle of August. Reef runs an internship program throughout the year. It runs for 3 to 4 months. I'm sure many of you are familiar with it, but if you're not, definitely swing by or hop on our website. And if you know somebody who is interested in marine science and just starting their career, we'd love to talk to them. All right. Now, I guess community announcements. There's a turtle dive coming up. What's going on with the turtle dive? Okay. It's June 23rd, right? I think so. Okay, June 23rd. Key Dives is doing a turtle talk and a night dive. Yeah. So if you're interested in turtles, hop on Key Dives website or give them a call, and they would love to talk to you about turtles and go on a night dive. Does anybody else have any community announcements? Anything happening? Cool. Look at that. It's July. It's in the Keys. Yeah. All right. So, well, now it's June. You're right. It's almost July. All right. So without further ado. Introducing Bobbie Renfro, also known as Sponge Bobbie, I've heard, which is awesome. And she will be talking to us about her work out planting sponges in the Florida Keys. So let's give her a warm welcome.
All right. How is my volume for everyone here and on the internet? Internet seems cool too? Great. I tend to project, so being mic'd is a little weird, right? So I'd like to talk to y'all today about the work that has been building up over the last 5 years for me during my PhD at FSU. Hopefully 1 more year left. And it's in partnership with Islamorada Conservation and Restoration Education, or iCare for short. Thanks for coming out. And it is the first ever reef sponge restoration program for the reef side of the Florida Keys. And to my knowledge, actually is the first ever reef sponge restoration program in the world, which is really exciting. And before we dive into what we're doing, I want to thank all the people we couldn't have done this without. Our funding sources have been through Moat Marine Laboratories' Protect Our Reefs program that is funded by those cool license plates you see with the corals on them around Florida. And then also the Florida's State Wildlife Grant Program, which is federal money given to the state of Florida that they get to divvy up to projects that they think will help the state's wildlife conserve and go on. And so we're excited to be picked for that too. And then I'm in the Wolf Lab at FSU. So Dr. Janie Wolf is my advisor. She's been studying sponges for 40 years. And I've learned a lot from her along the way. And I really appreciate my lab brainstorming a lot of the ideas sponge specific to the restoration project that we've put together. And of course, I have to thank Reef for letting me come here and share the weirdness of sponges with y'all. So I know probably some of you are familiar with iCare, some more than others, but I want to give a brief overview of the organization in case anyone doesn't know how the full scale of the project works. But iCare is committed to restoring the reefs of Islamorada, Florida, so our home reefs here. And the iCare Way to Restoration starts with year-long dedicated internships. So these interns, oh, it's y'all. These interns are spending a substantial amount of time down here in the Keys, understanding the ecosystem that they're working with, but also working hands-on day after day with the human community as well. And these interns provide education and engagement programs for recreational scuba divers to come out on a dive. Before the dive, they get to learn about the restoration activities of iCare, things that are threatening the reef and why we need to restore reefs. And they do a hands-on on-land demo of the restoration practices before they hit the water. Once the divers are all trained up, they get to go out into the water and actually outplant corals, sponges, and other organisms to the reefs of Islamorada, of course, with supervision from iCare's staff and from my FSU team for the sponge portion. And a lot of the organisms that they are putting out on the reef are housed in places like Islamorada's only on-land nursery, which was started by Moat in collaboration with iCare. And all of these organisms that iCare puts out on our reefs in Florida are then monitored by those interns. So we're not just putting them out there and hoping, I hope you do it. We're going back and checking on them to make sure that these organisms are healthy, that they're growing, that other, algae and things are not encroaching upon them. So it really is this sort of comprehensive both restoration and education program. And just to quickly touch on some of their coral successes, they've to date outplanted over 11,000 corals. They've done this with the help of over 2,000 recreational scuba divers and in collaboration with 4 of our local dive shops. And so these corals that they're putting out on the reef, right, that's the structure builder of the reef. We know the coral reef is named after the corals because they are the animal that is building up this whole 3-dimensional system that all these other organisms either want to live on top of or hide within or hunt around. But our corals cannot support this complex ecosystem all by themselves. They get to build the 3D structure, but they need other organisms to help them keep the ecosystem functioning as a whole. And I'll give you all one guess which organism I'm going to say helps them. Could it be sponges? It is. Corals need sponges on their reefs. So these wildly diverse, beautifully colored animals that come in every shape and size and color of the rainbow, they're one of the major helpers to the corals out on the reef. And it's definitely the brilliant colors of them that first caught my eye and made me interested in starting to study these animals for now the rest of my life. Actually, you heard about Roatan a little bit earlier. That's where I learned sponges first. They first caught my eye when I was a dive instructor there. And I kept having my, dive people that I was taking out onto the reef come back and say, wow, like the reefs of Roatan are so colorful. Like, what are all these colorful corals I'm seeing? And then they would show me pictures of sponges. And I'd be like, oh, whoa, yeah, you're right. Like a lot of that color really is coming from these guys. And that's what caught my eye and got me interested in studying them. But what are they? They're really strange, right? Does anyone have any idea? Is a sponge a plant, an animal, rock? Not a rock. So for a long time, people did think that they were plants. That was kind of originally where they started to be classified. And then we realized, okay, they're not plants. We figured that out, but then are they animals? We weren't sure. They had put them, below the animal tree of life at first. Finally, we figured out these strange creatures are actually animals, and they are the oldest of the animals. So here on this nice National Geographic infographic, thanks Nat Geo, you've got the animal tree of life sort of simplified. You've got us mammals up there. And down here, all the way at the bottom, are the sponges. So they're going to be the oldest of the animals on our planet, and we kind of all evolved from them. And the oldest sponge fossils that we've got floating around out there at this point are almost 900 million years old. So they've been on the planet a really long time. Anyone for context know how long dinosaurs were kicking around?
How long or how long ago?
How long ago? How long ago?
Well, the mass extinction of the late Cretaceous was 66 million.
That was such a good answer.
Birds are dinosaurs. Birds are the only living dinosaur that we have today.
Still kicking around. That was the best answer I could have hoped for. So as you just heard, dinosaurs, the last fossils of the dinosaurs are 65 million years old. The earliest fossils of the dinosaurs are only 250 million years old. So at 900 million years old, sponges are really beating the dinosaurs at oldest things on the planet, and they're still doing great. Dinosaurs, not so much. And so we've got our sponges that now present day are sort of this helper of the reef, but actually our ancient sponges had a hard skeleton like corals do today, and they were the reef builders of ancient times. So back, even before the dinosaurs, you didn't have coral reefs. You first saw a rise of sponge reefs, these reefs of sponges with these very dense, actually glass skeletons. And underneath, a very thin layer of sponge tissue was this hard glass structure that built these huge articulated reefs, just like our coral reefs of today. And between the corals of today and an organism called a diatom, which is a tiny plankton that has a glass shell, the diatom stole all the glass from the sponges and the corals stole all the space. So now sponges are sort of the assistant on the reef, but they used to be the reef builders. And what are they made of? Well, I just said glass, so we know that's in there somewhere. They have what you could consider bones, right? Our hard parts are our bones. They keep us rigid and upright. And the sponges have hard parts called spicules. They're typically on the sponges you're going to see here in the Keys made of tiny fragments of silica glass, but they can also be made of calcium carbonate like the corals on the reef. And you can see in the picture there that there's all these different, shapes to these spicules. You can have these stars, you can have, long hammers, you can have things that look like a curved sickle. And the different shapes of those spicules are actually one of the ways that we can definitively tell the species apart. So some of the species of sponges out on the reef look exactly identical to each other, but are completely different species. And so we'll actually take a small sample of their tissue and find all of the spicules in there, and we can use those to definitively name which species they are. And all of that spicules, all the little hard bony parts, are held together by the spongin fiber, which is a protein fiber. You can think of it like the ligaments in our body that are connecting our bones. This is a stretchy protein fiber inside the sponge. And you probably are most familiar with it in this form. So I brought a demo. I'll pass them around. These are bath sponges. And so our bath sponges, this was previously, an industry in the Florida Keys. It was kind of booming in the 1800s. It's a much smaller industry now. But the bath sponges are unique in the fact that they don't have any of the spicules in them. So that's why you can use them in the shower without getting covered in glass, which would be terrible. They're just made of this spongin fiber. So that's what makes a sponge spongy, are these fibers that you can squish in and out. And where can they live? Sponges can kind of live anywhere that there is water. Of course, I'm going to talk to you about reef sponges because that's where we're at. But they can even live in places like freshwater. So in North America, you'll see sponges like this green sort of little thin layer of slime hanging out underneath the rocks in your creeks. If you flip over rocks, you'll find either light green or light tan layers of sponge that are filtering the water in your creeks. In the Amazon, these are pictures of Amazonian trees here over the Amazon River. They probably have the weirdest of the freshwater sponges. So their freshwater sponges are about this big, and they're arboreal. They hang from the trees. And so during the wet season in the Amazon River is up in the canopy of the trees, which is great for these sponges. They're hanging out like it's a reef, but made of tree. But then the water drops 100 feet in the dry season, which would murder the sponges here in the Florida Keys. Your reef sponges do not like the air, but these tree-dwelling sponges in the Amazon developed a really kind of crazy way to get around this air exposure for 6 months out of the year. They roll all of their tissue, all their cells, all their DNA up into these microscopic little packets called a gemule. And the gemules are airproof. So then basically the sponge goes dormant in all of its little packets, all its gemules, until the water hits it again. They pop open and rebuild the sponge the next year. Super strange. Strange animals. They also live in mangroves. So we've got lots of beautiful mangrove sponges here in the Florida Keys. This is my favorite, the hot pink one. It's just brings so much color to the dark mangroves. But you'll find some really brilliant colors in the mangroves, and you'll see them always hanging from the mangrove roots underneath the trees. So if you say go kayak in Penny Camp, hop in the water or peek through the water, you'll see all the sponges hanging from the roots. And what they're doing down there is keeping other animals like crustaceans and snails from chewing away at those mangrove roots. So they not only clean the water in the mangrove creek, but they also help protect the mangrove from other organisms that would damage its roots. And they live out here in Florida Bay in our seagrass and hard bottom habitats. Hard bottom just means a habitat that has a limestone shelf underneath a thin layer of sand. So the sponges will actually kind of go through the sand and grab onto that limestone underneath. And when they're living out here in the hard bottom and seagrass habitats, these sponges are really kind of the only three-dimensional structure out there. So you'll get a lot of organisms like your juvenile spiny lobsters that are running around out in Florida Bay hiding underneath these sponges. A lot of fish hiding underneath the sponges. Lots of, air crabs and crustaceans tucking in there because it's one of the only places that they can find shelter from predators in this sort of open ecosystem, which is why they're super important in the bay as well.
So that in the top right is a sponge?
This one? Oh, sorry.
Yeah.
Right's hard for me when I'm facing you. Yes. Yeah, so this kind of dark patch here is going to be the sponge. It has a spaghetti worm on it, but this is a sponge that's actually one of the bath sponges. So this one, you could melt all the tissue out of it and get the skeleton, and it would be a bath sponge. They are more common, the ones that you can make a bath sponge out of in Florida Bay than on finding them on the reef side. So yeah. And then there are deep sea cold water sponges. They are some of the most strange looking of the sponge realm. This is the harp sponge. This individual was found off of California at about 11,000 feet under the sea. Yeah. And all of these other sponges, they make their living typically filtering things out of the water. That's how they're eating. But not this guy. This sponge takes its glass spicules and makes little hooks all along these sort of harp strings. And it waits for little teeny tiny fish or crustaceans to drift through and hooks them, and then kind of dissolves them like a spider and eats them that way. So it's considered one of the only sponges that's an active predator. It can't run around, but it's grabbing things. And down here in the bottom corner, we've got our glass sponge reefs that you're going to find in like your Arctic cold water areas. So these are very deep, very cold, dark water. And there's no, obviously, corals in these areas. So you again have these sponges making these huge reef structures, similar to a reef structure that you'd find here. And these are a little different than our sponges here as well, because they're all typically what we call a glass sponge, which is a sponge that has very little spongin fiber. It's all spicules glued together. And so they're very hard. Like you can't squish them. They're sort of like a glass vase underwater. I didn't put a picture of it, but it just came to my mind. There is a glass sponge that they give as a wedding present in Japan, because the Venus flower sponge, it builds this like glass cage that these two shrimp move into together, and they mate and they get trapped in there. And so it's eternal love. Dark take on eternal love. You're trapped in there. Do what?
See all the faces on there?
All the faces? Yeah, there are some different sort of eyes and mouths and things. Strange looking creatures. And so our sponges are living in all these places, but of course, I've come to tell you about the reef sponges because we are in the Florida Keys. And the reef sponges here do so many important roles for our reefs, keeping them functioning, keeping them healthy. One of the things they do is they'll act sort of as a glue. So they are gluing living corals onto the reef, and then they're also binding together broken up coral rubble. So if you have, say, a hurricane hit a reef, or you have a big wave crash down from a boat and pieces of the reef break off, sponges can help glue those back together. They also act as little living hotels. So you'll have crabs, you'll have shrimp, you'll have all these tiny, blennies and gobies and things that call the sponge their home. And then you have animals that like to eat the sponges. So your angelfish, they're big spongivores. They love eating sponges. Hawksbill sea turtles are another one that loves eating sponges. And then finally, they are the Brita filter of the reef. So we like to drink clean water in our homes. The reef likes to live in clean water, and the sponges are the ones that are able to do that efficiently. So there are other filter feeders out on the reef, but they cannot pull out the size of plankton that a sponge can. A sponge pulls out what we call the picoplankton. That's the tiniest plankton, the size of a bacteria cell. And all the other animals, they can filter the big things, big swimming zooplankton and stuff. But these tiny bacteria-sized plankton, the sponges are really the only ones that can remove that from the water column with any real efficiency. So let's dive a little bit more into some of these roles. How are they so good at filtering? Well, they have these really strange cells inside of them called a choanocyte. And that is this thing here. It's got the choanocyte cell body and then this little frilly collar and a little wiggly tail. And so the wiggly tail, the flagellum of the cell, spins around like a rotating tail inside of there, but there's tens of thousands of them doing it every little inch of the sponge, and they create this massive suction that pulls water into the sponge. So people used to think that sponges were like, oh, I hope a wave hits me or I'll never get to eat. But now we know that they actually have a lot of agency over their eating. They can speed up their filtration. They can shut down their filtration. And they all do that by controlling these cells. And then while the water is getting pulled through, the frilly collar on the cell, this part here, is sort of like a fine net. And it's scooping out of the water all of those bacteria-sized plankton that then the choanocyte cell sucks into its cell body and decides, do I want to eat you or do I want to spit you back out because you're toxic or gross or something? So they even can choose what they want to eat out of the water, which is something that we're still just barely learning about now. So that's pretty new information that we've gotten from the sponges. And of course, them being the filter of the reef is very important. We want crystal clear water to dive in throughout the Florida Keys and the Caribbean. But one of the jobs that they play that is even more important with respect to restoration is that role of being the glue of the reef. So we want a reef that is holding together, that is resistant to erosion, with corals attached. And the sponges are going to be really helpful in doing that job. So here we have a sponge, the orange part. That sponge is the orange icing sponge. If you like scientific names, its name is Mycale laevis. And it's underneath our mustard hill coral here, our Porites astraeoides. And so you'll see these two growing together a lot on our Florida reefs. So they call it the icing sponge because it looks like, cake icing that you just scooped along the edge of the coral. But you can see that both the coral and the sponge have altered their bodies to grow together real tightly. So, that coral isn't typically quite so lacy looking by itself. This sponge, if it grows by itself, grows as a ball. But together they make this shape. So they're really adapted to growing with one another. The sponge gets to kind of be protected up underneath that coral so things don't want to eat it. And the coral gets this extra layer of adhesion, this glue that's helping it stay down onto the reef.
Which came first?
I don't know, actually. That's a great question. Yeah, I'm not sure which came first. It can do this with other corals, so maybe the sponge came first, but I'm not sure. And we see a lot of sponges and corals next to each other on the reef, but they're not all a known, symbiotic relationship like this. Some of them are just two strangers next to each other on the reef. So let's look at a pair that's like that. So this burnt orange sponge, I don't have a common name for it, but its scientific name is Ectioplasia fairhox. and this is one of your lettuce corals, an agaricea. And you can see that they're growing, right up against each other on the reef. So if you were out diving and you saw these two so close to each other, what would you think was going on with those two animals? I heard competition whispered out there. Yeah, a lot of times we think on the reef, if two animals are really close to each other, they must be competing for space. But let's zoom in and see if they look like competition is happening. So if competition is happening, you would think they're going to look unhealthy. They're going to look like they're fighting. That edge is going to be, eroding from one animal because one is winning at the fight, right? But here we see nice, healthy orange tissue all along the edge of the sponge that's touching the coral. We also see along the edge of the coral here, these are the polyps that are nearest to the sponge. And they're all whole. They're all whole polyps. There's no half polyps getting cut off by the sponge. There's no polyps of the coral that are dying back because they don't like to be near the sponge. So you have two healthy animals growing right next to each other. You can see that the coral is sort of going up over the sponge a little bit. But the neat thing about sponges is that they can rearrange their bodies. If I was it'd be cool to see out of the back of my head. If I was a sponge, I'd just take one of my eyes and put it back there. We can't do that, but they can. They can move things around on their body if it's inconveniently placed for them. So for instance, this little hole you see in the sponge, that's one of the places where water goes out. And that excurrent pore on the sponge where water goes out, it doesn't want that covered. That would be bad. But if the coral grows over it, it will just scoot it somewhere else. So they're really good at living up close and personal with corals without the coral causing the sponge a problem, but also without the sponge causing the coral a problem. And that's really great because that allows both of them to have more attachment to the reef. But in particular, it's helpful to the coral because the coral is, a hard, rocky type animal glued to a hard, rocky reef. There's not a whole lot of shock absorption there, say, if you've got a big wave that hits you. Whereas the sponge being squishy, it can give a little bit. So maybe the coral gets knocked off the reef, but the sponge is able to hold it on. And my advisor, Dr. Janie Wolf at Florida State University, back in 1979, was kind of revolutionary in trying to prove that this was true. So she thought it was true. She was seeing sponges and corals growing next to each other. She thought, I think they're helping each other. I don't think it's competition. How can I show that? So she did something that no one would give you a permit to do today. She went to all these patch reefs in Panama about the size of, your dining table, little reefs. And on some of them, she left them alone. And on others, she peeled every single sponge off. And I'm like, wow, the science you could do when nobody was watching. Cannot do stuff like that anymore. And she peeled all these sponges off these little reefs. And then she gave it 6 months and came back to look at them. And she saw that on the reefs where She left everything alone. Sponges and corals are just like they're supposed to be. About 4% of the corals had popped off the reef just by natural chance. You know, maybe there was a storm, maybe another fish hit them or a boat anchor or something. On the reefs where she had taken all of the sponges off, 40% of the corals had popped off the reef and were still below the reef, rolling around and dying in the sand. Not so good for those reefs, but really cool for science. Because it really showed definitively how much the sponges are helping the corals stay adhered to the reef. So with your sponges on the reef, your corals survive 10 times better, which is pretty amazing. And you can see here, we've got one of iCare's coral outplants, our staghorn coral here. And this sponge just naturally grew up through it. We didn't put it there, it just did it on its own. The coral looks healthy, happy, right? Nice orange tissue. And now the sponge, it's getting two things out of this. It's getting to be supported a little higher in the water, which it likes because it can filter better. But it's also helping the coral stay on the reef because you can see the sponge has touched down here. The sponge has touched down here. Hard to see in the picture, but back here, the sponge is touching the reef again. So the coral goes from 1 base on the reef to 4. So if the coral gets knocked off by a storm, the sponge can help it stay in place. And it not only helps the living corals, it also is a friend of the dead. The sponges are really helpful to all of the dead coral rubble that's out there on the reef, because you'll have these areas where you get coral rubble built up and it could become new reef. It could become a place for, new corals to land and grow. But if it's loose rolling around, they can't stay there, right? They're going to get rolled into the sand. The new corals won't be able to make it. So we need that rubble to get glued back together somehow. Now, coral and algae, some of the other animals on the reef, they can do it, but it's usually a slow process. This study that I have a graph from here was by Biggs and done in 2013. And Dr. Biggs went out and had all these rubble piles. Some of them, he just left them alone, and some of them he added sponges to. So you've got a sponge added here, 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 here. And so he put the sponges in the rubble piles, and then he stepped back and waited to see what would happen. And the first thing he saw was that, looking at our graph here, this is number of rubble piles. So we had a whole bunch of that little setup, some with sponges, some without. And these are the ones with no sponge. These are the ones with sponge. And he checked on them at 12, 21, 33, and 45 months. Now, if he has a gray bar on his figure, that means that the rubble's still loose rolling around. Anywhere that the gray bar turns black, that rubble has now become consolidated and it's a stable substrate for corals to live on. And he found, first of all, that you got to stabilize a lot faster if you had sponges. So it took 33 months for the loose rubble to become a stable substrate if it didn't have sponges, but it only took 21 months if you got sponges added to your pile. He also found that by the end of the experiment, at 45 months, more of the piles that had sponges in them were a stable substrate that a coral could live on, versus this little bar here for the rubble piles that had to do it all on their own without the help of sponges. And probably the thing that I thought was coolest about this experiment is that he found out it's not just about the stability, because next step, he stabilized some of the piles with concrete. So this is a little blurry, but here's the rubble. And in between, he has laid concrete. So now he has instantaneously made it stable, right? You'd think, coral would love to land there. Well, what he found is that even then, even though this is completely stable, sponges plus rubble still attracted more baby corals to land and grow. So something not only about that rubble being still and locked down to the bottom, but also about the sponges being there told the baby corals, that's a place I want to live. So I thought that was pretty awesome that, the sponges also might be kind of a cue saying, hey, corals, it's safe over here. And here's an example of this activity from our home reefs here in the Keys. So this is 2 pictures. Both pictures have this staghorn coral in them, and both pictures have the green finger sponge in them. The green finger sponge is one of the ones that we work with in our restoration projects. So on that panel over there, that photo is a natural staghorn coral that was growing at one of my research sites, and it got broken off the reef. I don't know how, but I saw it and I was like, oh, you got broken off the reef. And so I kind of set it back on top of the reef. I was like, find a place that you're safe. And it rolled down and got caught by the green finger sponge. And I thought, very cool. All right, my sponge is helping this coral. So natural sponge, natural coral. Now looking at this picture, this is one of iCare's restoration corals, and this is one of our restoration sponges. And this restoration coral got knocked from its little group and rolled across the reef and got caught by one of our restored sponges and held back onto the reef. We're doing it! It's just like nature. This was, one of the moments underwater where I was like, through my mask, because it was so cool that this was so similar to what I had seen happening naturally out on the reef. And so that's the goal that we're going for. We're trying to help the reef hold together, and we're also trying to give more things on the reef that will help hold the corals down. And the sponges are perfect for both of those jobs. So I hope I've convinced you that sponges help restoration and help the reef, but do they themselves need to be restored? Well, the answer is complicated, like all things in science. For some species, absolutely yes, and for some species, no. So this sponge that's pictured on the slide here, it's an excavating sponge. Its name is Cleona dilatrix. Cleona, it likes to dig into the coral rock, not good for the reef. Normally, it's in pretty low numbers on reefs. That is a native species here to the Florida Keys. But this sponge loves pollution. You give it some sewage, and it's like, yes, I'm going to grow like wildfire. So it loves sewage pollution. It's a sponge that does not need to be restored, one, because it doesn't do a good job on the reef, and two, because it is one sponge that actually is doing better with human disturbance to the ecosystem. But this is a very rare case. So the majority of sponges, like way more, this is a handful of species versus thousands of species that do all of the really good jobs I talked about on the reef. And a lot of those sponges, like our giant barrel sponges, they're having problems out on the reef. So this here is a giant barrel sponge from one of our restoration sites that has experienced a disease. It hit all of the giant barrels on this one reef. We think a lot about stony coral tissue loss disease hurting our corals. Sponges have similar threats to them as well. So this is a wasting disease that actually causes them to melt. So you can see where the top would have been on the sponge, and it's melted down to just this much left of the barrel. So sad. But that's, one of the things that's telling us that we do need to act now and act quickly on learning how to do coral and sponge restoration together, so that we're helping these animals in case this disease becomes on the scale of something like stony coral tissue loss disease.
Horrible diving, and the boat anchored, our boat anchored, and just decapitated a bunch of these big barrel sponges. But one of the scientists on board went down and was placing the decapitations back on, and was telling us that it'll grow back together. But this is disease.
This is disease.
They don't have any.
No, after a lot of time, they will rebuild if they can, depending on how much of them is left. They want to be in the barrel shape, so they'll try to regrow that barrel shape up. With ones that are knocked off, say by like an anchor line, if you can match the top to the bottom, they will fuse back together because they know self, and they'll fuse. But you have to get them paired up right, and you have to somehow help them stay on. So like run some zip ties through them or something would be a solution. But yeah, unfortunately, this is from disease. So it's a little harder for them to come back from. The other thing you could do with something like that is just find a way to wedge it down into the reef, and it'll grab on and start growing itself from there as well.
Separate from it.
Mm-hmm.
So just keeping it.
Yep, just keeping it down. The top of it will realize the reef is under it and try to hold on to it.
Are most of these diseases that are attacking both the corals and the sponges viral? Because if the sponges filter out bacteria, are they helping the reef in terms of cleaning up bacteria as well?
That is something that someone in the room sitting directly next to you is trying to learn. Everyone, Josh Farmer. Josh, I'm breaking the clicker. Josh is trying to learn that for us. So hopefully we will know that answer soon. But we don't know right now. And yeah, next talk. And so we have disease in our sponges. We also have just general losses of sponges in the way that you're used to seeing losses of corals with changing ocean conditions. So we know our reefs and our oceans are getting warmer, they're getting more acidic, they're having increases in storm frequency, increases in pollution. And so in a lot of areas, you're seeing figures from our studies of monitoring populations of sponges that look like this one here on the slide. So to orient you, we've got this was done in tissue volume of sponges, but basically like how much sponge is out on the reef. And then we've got years across the bottom. So from 1984 to 1998 on some reefs in Panama. And what we can see is that from 1984 to 1998, we have a loss of the general sponge community at these reefs. In this case, for this paper, they thought perhaps it was due to nutrient pollution, but they didn't have the data to show a correlation. But in general, we saw a decline. We've also seen these declines in reefs in Belize, but these declines are really hard to capture because there's not a lot of people taking species level data of sponges. So you'll have something like a monitoring project that monitors everything as sponge, right? Which is easier, especially if you don't know them. There are so many. But then you can have something like Cleonodilatrix, that orange sponge that digs away at the reef, making it look like sponges are going up, when really only it's going up and everybody else is going down. So my lab, we're one of the places that we're trying to really put a lot of legwork into having these long-term data sets so that we can see what's happening to all of the sponges out there, both together and separately as individual species. And one thing that we have seen affecting the sponges here in the Florida Keys is hurricane damage. So we know that we're seeing increasing both frequency and severity of tropical storms around the world, which is correlated with changes in our global climate. And one of the results of that is that areas like reefs are getting hit with these severe storms more often than that system is used to recovering from that activity. So hurricanes are natural, right? They've always happened, but they're starting to happen more. And when they happen too close together, the reef animals don't have time to bounce back from it. And so what we've seen here, we have pie charts on top that were before Hurricane Irma and pie charts on the bottom that are after Hurricane Irma. And the red wedges, those are going to be your sponges. So you can see both in the middle keys and the upper keys, your red wedge is shrinking after Hurricane Irma. And what they found was all of these sponges after the hurricane just rolling across the sand because the sheer wave action of the hurricane, being over top of the sea was just scraping them away off into the sand. And they would recover naturally on their own, but it would take them a very long time. So we still see areas both in upper and middle keys and down at Looe Key, where the sponge communities have not bounced back. And we know Hurricane Irma has been many years now past. So that speed of recovery is very slow. And that's one of the things that we're trying to combat with restoration is the fact that, sure, the reef would probably get its sponges back at some point, but would there be any reef rock left for the sponges to come back to? Because in the meantime, that thing that's protecting the reef from erosion, all those sponges, isn't there to protect the reef from erosion. And so we're still getting a breakdown of the reef without the animals that are trying to stop that. So we're trying to help them move just a little farther, a little faster with some restoration to where they would have ended up on their own anyway. And anchor damage is also a problem. So these are ones that have been sheared off by anchor damage. And of course, the Keys is working on this problem by having things like mooring buoys installed into the popular dive site areas, and that's awesome. But accidents do still happen. So these are two sponges where you can see it's not melted in like that other picture we saw. It's a real clean shear across them. And that's how you know that anchor damage has happened in an area. With these sponges, because the base is still here, the sponge may actually be able to recover from this, but it will take a very long time for, this much pancake left to grow a sponge this tall. So working on those kind of damages is important as well. And there is a lot of work on sponges both on the reefs and in Florida Bay. So Florida Bay has these beautiful sponges I talked about that the spiny lobsters like to hide under that create all of this, three-dimensional structure out in Florida Bay that also drive our bass sponge fishery as well. And these sponges had a mass mortality event in the 1990s that was related to a big bloom of a harmful algal bloom. And they really have not recovered in Florida Bay ever since that harmful algal bloom in the 90s. So you can see how slow sponges can be to recover from damage. And FWC took charge on that and have a sponge nursery where they've been going out and putting those bay sponges back into Florida Bay. And that was one of the things that actually inspired iCare and I to think, whoa, look at what they're doing for the sponges out in the bay. We should be doing that for the sponges on the reefs. But we couldn't necessarily just use their methods because their methods were designed for this shallow, low wave action, flat environment. And we have a high wave action, three-dimensional structure that we want to put sponges on out on the reef side. So we had to develop our whole new program specific to the sponges that live on the reefs. And so what we wanted to do is create not only a restoration, but an education program in following with the iCare way. We started out by determining if it's sustainable for us to collect little fragments of sponges from healthy reefs and move them to degraded reefs, which is how a lot of coral restoration started. It was little clippings of corals, kind of like your gardening, before we developed methods for creating nurseries and things like that. So we wanted to see, can we do that with the sponges? Then we needed to design methods to put the sponges out on those reefs and to see if once they're out on the reef, do our methods hold? Are they going to last? Once we designed methods, we wanted to educate and engage recreational divers and get them involved in the activity of putting these sponges out on the reef. I even see some of our divers in here that help us every week, which is fun. And then we wanted to monitor those animals. So not just put them out on the reef, but make sure that 6 months, a year, 2 years later, these animals are still looking healthy and still, on the reef where we have placed them and are growing. And we have focused a lot of our attention on alligator reef for the first 2 years of our work. This 2023 summer is the first time that we are branching out to a new reef, but most of the data that I'm going to show you comes from restoring sponges to the famous alligator reef. I've got the lighthouse here. I don't need to sell everyone in the room on the popularity of this site, but I have this slide in for talks not in Florida where they're like, what's so special about this reef? It's a very popular place and we want it to stay healthy. As you know, it's pretty shallow. So you're looking at, 20, 22 feet in the sand if you dig yourself a little nest. And we've got mooring balls running all along the side of that reef. So reducing some of that anchor damage. And you've got a reef shelf running along that's, somewhere between 16 to maybe 19 feet underneath the water. And we have 3 sponges that we are working with in our restoration. Our first sponge is Aploxyna coliformis, but we can call it the purple rope sponge. We have Amphimedon compressa, which you can call red branching sponge, and Iatricota biricillata, which is the green finger sponge. And the thing that these 3 sponges all share is that they all have a branching shape to them. So you can see they're all... Oh, I guess a big truck went by. So you can see that they all have this branching shape to them. They're all tangled up with each other. And so these branching sponges, one thing that they like to do is they grow in and around animals. They grow in and around rocks. They're used to that. So they're really good at holding stuff together. And they only touch things in little places, which is super helpful if you're wanting to hold a coral onto the reef. You don't want a sponge that's grabbing it all over the place. You want a sponge that'll just, oh, hi, coral, I'll just touch you a little bit there, and then I'll touch the reef, and I'll hold us all together. And that's what these are really good at. They also are very good at what we call asexual fragmentation. So that means that naturally, like without any help from humans, these sponges will break apart in a big wave or in a storm, and little bits of them will roll to another part of the reef and grow the same sponge at a new part of the reef. So we are doing what they would do naturally. We're just helping them go a little farther than they might roll in the next 10 years. And so we wanted to see, we don't have a place to collect these from as a nursery. We have to get them from other animals on the reef. So can we do that sustainably? So we went out. You can see one of my undergraduate students from Florida State here. She's measuring little pieces of sponge. We collected 10-centimeter pieces about this big of the ends of the branches of the sponges, and those were going to be our restoration sponges. And then you can see here in this picture, this red branching sponge, she removed the top of it and then tagged it with some beads so that we could go back 3 months later and measure to see how much that sponge had regrown that tissue that we took off, to see if we take this tissue from you, are you still okay? Will you grow fine on your reef? And we found out that, yeah, actually at the current operating scale of iCare, So we don't visit these reefs particularly often. We're collecting them, just in the summer and then leaving them alone the rest of the year. Current operating scale, we are sustainable. So these sponges are able to regrow between 4 and 6.5 months, all of the tissue that we collected from them. And so obviously if we want to expand, we want to start looking into things like maybe creating sponge nurseries. But for our current operating scale, these sponges are regrowing the tissue twice as fast as we're collecting it from them. I don't know how fast the starfish goes, but they're pretty fast too with the arms. Yeah. And these numbers that we got are reflected in other scientific papers. So we're not the only ones that have tried to test how fast these animals can regrow their branches. And other people found the same thing that we found, that they can regrow them in, roughly 4 months to half a year. The other thing that I want to note about this is we only measured that one branch we collected, but the sponge can grow in any direction. And so it could also be that, they grew back that branch in 4 months, but they grew 5 other branches over there also. So it's a conservative estimate. So we know that we can do this sustainably. Then we had to figure out how are we going to put them back on the reef? And we did that with this method that it took some iterations to get here, but this is what we landed on. We take donated pieces of staghorn skeleton or rubble from the reef. So we have a permit to collect rubble from the sand that's loose rolling around. But we also have a lot of restoration partners that have these staghorn skeletons left over from, say, a group of restoration corals that got diseased in the tank, and they decided we're not going to put those on the reef. Well, this is a protected species. They can't do anything with those skeletons, so they just sit around. And I said, why don't you try to use them for this? We bleached them, dried them, left them out in the sun for a very long time, make sure that they are clean. And then we attach the sponges to them with these thin beaded cable ties. And we have the sponge here on top of the rubble, and then we clear out a little space on the reef with some brushes so that we can get all the algae out of the way. And we mix up some underwater epoxy. It's the aquarium safe type epoxy that doesn't harm any animals, has nothing weird that will leach out of it. And we put that on the coral rubble, but not on the sponge, because sponges, they can be a little fragile. If you put epoxy on it, it'd be like, oh, epoxy on me. I'm going to ditch that end of myself, which would not be helpful for it staying on the reef. So we epoxy the coral rubble down. And what will happen is this sponge is going to grab onto this piece of coral rubble. And you can see here that this image does not have any of those little cable ties in it any longer. Once that sponge has grabbed onto the rubble, we go back out 1 to 2 weeks later and we clip off the little nylon cable ties, and you just have sponge, rubble, and epoxy, which is similar to concrete, pretty natural out in the environment. So we're not leaving any plastic out there. The epoxy gets covered over by crustose coralline algae and other algae within 1 or 2 months, which makes them look super natural in the environment. Like you can't differentiate them from the natural sponges, which was so cool to see at first until I realized I had to find them again. We're tagging them now. I've learned a lot. And so these are sort of large adult sizes of the sponges that we are looking at. So you can see that we're collecting, this much of this whole individual animal. And over the course of summers 2021, 2022, and now currently in 2023, we are up to 825 sponges that we've put out primarily at Alligator, but now at another reef named Captain Arnos. And so we are tracking these sponges for their growth and making sure that they're doing well out on the reef. And we've learned a lot from putting all of these sponges out on the reef. We picked our favorite brand of epoxy after getting mad at several others. This one comes in a pre-measured stick form. It's almost dummy proof, almost. And we also found that it sets faster than some of the other ones. And the sponges are sort of like, they're real good at catching the wave action because they're up on their piece of coral rubble. And so if the epoxy doesn't harden quick enough, they pop right off. We also found out that a recreational diver during a 45-minute dive can confidently transplant about 5 sponges. So we tried more, we tried less, and we came to this number as the ideal. Upright is not right. They do grow that way, but they don't want to be transplanted that way. We found that when we transplant them in an upright position, it makes them even more likely to catch wave action and get knocked off. So we'd come back out after a storm came through and all our sponges are on the ground. We'd stick them all back on. And so we are started laying them down flat on the reef. And what they'll do is they'll grab onto the reef first and then turn themselves up. So they're getting more secure by grabbing the reef, and then they'll right their orientation. And of course, like everything, storms are bad for our restoration sponges. So we would not want to outplant directly before a storm, but more importantly, if a storm does come through, we go back and check on all those sponges, resecure ones that are loose. iCare's awesome at like contacting me when things like that happen, so that Kylie can update me on what the sponges look like and ask me what we should do. So that's been really great when I'm still up at school. And so these are some of the major things that we have come away with so far, and we're continuing to test and develop methods to really try to hone in on what is the best practice for this. And through this program, of course, we also wanted to educate and engage recreational divers, because that's half of iCare's mission. These are the people that are out on the reef. They're the ones enjoying and loving the reef and caring about the reef. And a lot of times you also want to give back to the reef. And so this is a way to do that. And we do that through our courses on sponge restoration and coral reef sort of sustainability. So learning about things like, what are the threats to reefs? What are things that you can do to help the reef in your daily life? And then also learning why the sponges are important to the reef and how to restore them. And then we do a hands-on on-land demo. So practice everything where you can ask questions without going underwater. You can use verbal cues. And then we're able to sort of get really honed in on those methods on land before we go underwater. And then in small supervised groups underwater, our divers are putting out these sponges on the reef with our help. And then we're able to go back and monitor those sponges later, because we're now putting tags at each location to make them a little easier to find. You can come. You can do this. We have a few trips left for the summer, so it's every Wednesday until July 5th. And if you call Key Dives, that's the shop we're currently running the sponge restoration program out of. So if you call Key Dives and ask to go on a Wednesday sponge dive, they can get you all signed up. It's a class in the morning starting at 8:30 at the barn behind Key Dives at Bud and Mary's. And we talk about all those cool things about sponges. It's some things you've heard here and some things that you haven't, so it's not totally the same talk. And then we go out on a 2-tank afternoon dive, and you get to put sponges out on the reef. And through this education and engagement program, we have given 3 FSU undergraduate research assistants the opportunity to get critical hands-on career training, both with working with the public in education about, the environment and restoration, and then also through being able to do that scientific monitoring, working with the data, and being able to have sort of this, I think, a more comprehensive experience with being a scientist than maybe you typically get in college, because we're not only doing the science part, but we're also doing the communication part that really is so critical to science. And I should have put up here also one University of New Hampshire undergraduate research assistant. Everyone, this is Jack. And he is down helping us with the project this year from his university as well, which has been really exciting. And these students have helped conduct 15 of these sponge restoration courses, through which we have engaged and involved 108 divers in our restoration. So once we got all those sponges out on the reef with the help of our volunteer divers, we wanted to go back and check on them. And we did this using photo data. So there's so many of them out there, right? I can't sit and measure them all by hand. So we go up to them with a ruler and a camera, and we take a photo just like you see here. So this is the sponge in the photo. And actually this little bit here is the only bit of that coral rubble left visible. So it's growing around it, kind of using it as a fake skeleton for the sponge. And we have the ruler in the photo, because this program up here called ImageJ, it's free online if you need to measure anything in your pictures. It's a free software that a lot of biologists use to measure animals from photos. You just have to have a ruler in there, and you can scale the photo based on the ruler. And then you can see a real thin yellow line is drawn across the picture. So I draw those lines in different directions to get different measurements of the sponge as it changes width throughout the picture. And then I just use good old geometry from 6th grade to figure out roughly what shape I think the sponge is, typically cylinders, right, because they're branches, and use those measurements to calculate the volume of tissue that is there to see how much that tissue volume is increasing, and thus how much are they growing. We've checked on them at 0 and 6 months, and this summer we'll be doing our first 1-year checkup on the 2022 sponges. And so some of the data that we have come out with so far, our survivor data, we have had from 2021, our sort of kind of small scale pilot study where we're still trying to develop a lot of the methods, learning a lot of those things we did wrong at first. We had a 45% survival, which I was like, oh, I wanted it to be higher. But then a lot of coral restoration people told me for a first year, like, that's pretty good. I felt better. And then in 2022, we went up to 49.3%. But those 365 sponges in 2022 also went through Hurricane Ian and a benthic cyanobacterial bloom. So they had a harder road than the 21 sponges and still survived better, which I was really excited about because I think it says a lot about us honing in these methods, and we're going to continue to experiment with them to really get it perfect, which has been a fun experiment. And here we have colored bars to match our sponges. So our green finger sponge is the green bar, thin purple rope sponge is the purple bar, red bar is the red sponge. The gray area is the sponges that were missing when we came back 6 months later. The colorful area is the sponges that were still there. And so one thing you can see from this picture, and it's easy in the picture, but we also tested it with some statistics just to make sure. But the species is not independent of its survival. What that means is this test called a G test that tests if two things are independent of each other. We saw, does it matter what species you are? Do you always survive 49% of the time? Or does one species survive better and one survives worse? And what we found is that green finger sponge, even with more of them being outplanted as a whole, it still had much better survivorship than the other two species. And in particular, you can see down here, the Amphimeton compressa had the lowest survivorship. That red sponge had the lowest survivorship. And so some of the things that we're learning from that are things like maybe some of those species are less tolerant of how we're transporting them. Maybe some of those species don't like alligator. Maybe it's just the site. So we're expanding sites. We're testing different methods of moving them around to try to see how we can get those numbers to go up for all of those species. Even though we did lose individuals from each species, overall, we had an increase in our tissue volume. So going back to those photos in that program and measuring how much they had grown after 6 months, all 3 species grew in that 6-month period. And so you can see that even Amphimeton compressa, which had pretty low survivorship, still grew a lot for the individuals that were left on the reef. About the same as its neighbor, Iatricota, the green finger sponge. And then the purple rope sponge looks like it didn't do as well, but it's much skinnier than the other two. So a small increase in its tissue volume is really a big increase in the amount of branches that it has. So this is a summary of everything that we've done, but essentially we've come up with these restoration methods for the first reef sponge restoration project in the Keys. We have engaged 108 divers to restore 825 sponges. We have found that our green sponges are doing the best so far, and we're in our 3rd year of restoration, and we're starting to contemplate ideas about trying to build a nursery so that maybe we can pull sponges from there instead of from natural populations. So that's my contact info if you want to say hi. Any questions?
It looks like you have a distance between each one that you're planting them with the epoxy. It's like a foot or something like that, or it's measured.
Yes. So
when second part to the question is if you're outplanting with coral outplantings as well, do you keep that same distance, or can you plant them even closer to a new coral?
So that's kind of the next thing we want to start experimenting with. That is tied in again to Josh's project. He's looking a little bit about distance from the corals to the sponges. But at first, we just kept them separated just to see how the sponges would do on their own. But we have started to talk about trying to experiment with what if we put them close together? Do they both do better and things like that? So that's the next phase. Yeah.
Do sponges have any similarities to like the coral with making them super small is called microfragmentation.
Oh, yeah. Could we microfrag the sponges?
Yeah.
I don't know yet. I think it's going to be really dependent on the species. So like that thin purple species, it's pretty easy to cut it into really small pieces and they do pretty well just like that. But the thicker red ones, they're real sensitive about if you like try to cut them too small, they spit all their cells out into the water. Not ideal. So that's something I'd love to test out. I don't know yet, but it'd be really cool.
Are microplastics a problem with the sponges?
There have been studies that have found that microplastics may concentrate sponges, so pull them out of the water and keep them inside the sponge, which some people have suggested is another motivation to restoring sponges. I think my only hesitation to be a proponent of that aspect is the fact that if the sponge dies, it's going to just release all the microplastics again. So it's really not a good permanent solution to microplastics, but so far it doesn't look like the microplastics bother the filtration system, which was something that we were wondering about. My lab didn't test it, but another lab did feed small microplastic beads to sponges, and it seems that it didn't change their filtering ability. So hopefully they're not getting stuck in there at all.
Maybe with the Japanese glass sponge.
Yeah, get stuck in that one like the poor shrimp.
You mentioned in the beginning that they can shut off their filter, their suction. When would they want to do something like that?
Yeah, so they will do that if they think the water around them has gotten strange for some reason. So like if you put them in a bucket and they get too warm, then you'll see that they'll close up their pores. I'm trying to see. This photo isn't a great example of it, but here's a pore here. You can see some down here. And you can see how you can see into them. It's a dark hole into the sponge. They'll actually shut it. So when we are restoring them, they don't love the boat ride, and so they'll all close. And then as soon as you get them on the reef, they open back up again. I think another one of the aspects to that may be that when they're stressed, they're trying to save energy, so they shut down their pumping system just to like maintain until better water comes or better times come. But yeah, it's pretty neat. Like once you get used to seeing it, you can see like, oh, you're not pumping. Oh, but you are.
Do sponges produce or reproduce sexually as well, or just asexually?
They do, and there's not a whole lot known about that. So we know that a lot of them do. The green finger sponge that we have in the restoration project is one that has really big larvae that are like a bright kind of, I think they're like a bluish color. So if you cut them open, you can sometimes see that they have larvae brooding inside of them. But a lot of the sponges, it's kind of a mystery to us still how they asexually and sexually reproduce. The barrel sponges are one here that put on quite a show, kind of like a coral. They'll look like they're smoking. And so those are ones where I've never gotten to see it. I'd love to see it. But they'll actually emit their gametes, their sperm and eggs into the water the same way that the corals do.
Are there fire sponges?
There are. There are in the Keys, and they do burn. The fire sponges generally live either in the seagrass or hanging from the mangrove roots. So if you're in the seagrass or the mangroves and you see a very bright red sponge, you just leave it alone. It will burn you. It kind of is like fire coral. It goes away pretty quickly. There is another sponge called the touch-me-not sponge that I've never seen in the Keys. I haven't seen it here, but it gives you more of like an allergic reaction type rash. And for most people, it's pretty innocuous, but some people are allergic to it. But I haven't seen them here, so hopefully they're not.
Do you know if the resiliencies and sensitivities of corals and sponges like complement each other, or do they align? Like is coral like, okay, I got this, and then sometimes sponge is like, okay, I got this.
So I think with physical damage, we know that the sponges do a little bit better of tolerating some physical damage, but with temperature, we're still learning whether or not sponges are okay with that. So we don't really know there. We do know that with ocean acidification, a lot of the glass sponge, or not glass sponges, but the sponges that have the glass spicules in them are probably less affected than the corals because it's not changing that, they don't have any calcium in them. There are sponges with calcium spicules in them, and one of my friends actually just found out, she just published it, so exciting, that those sponges with the calcium spicules have a sheath to cover the spicule up so that they actually don't get affected by ocean acidification the way that the corals do. So yeah, pretty wild, yeah.
So is this individual sponge, is that one animal or is it a colony of animals? And how does it communicate between cells? I mean, how does it make the pores close up? And where is its brain?
That is such a good list of questions. Yeah, they're all connected. Where's its brain? How does it communicate? Is it one animal? So corals, are colonial. They have individual polyps of the same genotype, same individual, but different, little animals all living together in a colony. Sponges we call clonal, which means that like each cell in the sponge is sort of a clone of all the other cells, but they kind of can act independently. So it's a lot more like a bunch of people in a city deciding to all work together. And so the cells in the sponge, like a stem cell in people, they can reassign their jobs whenever they feel like. So you might be a cell that runs around cleaning up the debris in the sponge one day, and then there's too many of those, so you switch over and decide you're a cell that makes the spicules now. So they can all kind of reassign jobs. The communication is mostly through chemical cues to our understanding. They don't have any sort of centralized nervous system. So no brain, it's all cell to cell, but you'll see certain things like you can see certain signals propagate through the sponge. So like if you touch one end and it closes the pores on that end, then they'll kind of go down the sponge and close. There's some sponges that will retract from you when you touch them. They'll shrink up, and that you can kind of see ripple across them, like the part you touched will contract first and then the rest will follow. So they are able to communicate across their cells, but it's very different than how any of the other animals work. You could also put them in a blender, and then you could pour this juice into a little dish with some seawater, and all of these cells would go, whew, we're not in a sponge anymore. Everyone get rid of your jobs. Sink to the bottom. They'll all go to the bottom and climb toward each other and start rebuilding a sponge. They'll all assign new jobs. It's very strange.
How long does that process take of rebuilding?
It takes about 24 hours for them to start forming very small little groups. And then the small little groups over the course of like a week will find each other and start building up a taller little sponge. Yeah.
What are the differences between tunicates and sponges?
Ooh, okay. So tunicates have actual tissue layers, like how we have like a epidermis, a skin, and we have organs. Tunicates have organization inside their bodies like that, where sponges don't have any plane of symmetry, and they don't have any like true organization into tissue. So they have an outside layer that's different from the middle, but it's one cell thick, which doesn't meet the definition of a tissue. So they're just sort of, they're very simple, but in an elegant way. I think David's trying to kick me off the stage.
All right. Well, thank you so much for coming, Bobbie. We really appreciate it.
Cool.
It sounds like Bobbie's in the queue till July 5th.
Yes,
Yeah.
I am.
So we'll have our next Fishing Friends July 11th. But if you would like to get involved, sounds like call Key Dives.
Call Key Dives, come out sponge. Shoot me an email if you have more questions about sponges.
All right.
How soon will this be posted?
How soon will the recording be posted? It's usually